Karl, how's it going? Nice to see you again.
Oh, great. Great to see you too.
Well, thanks for taking the time to talk to us here at Neuropsych. We want to know what you've been up to, and talk about the paper that you had come out recently.
Sure. So we got this paper in molecular psychiatry, which we were really excited about, and we're all very happy about how it went. So maybe just a little bit about myself first. So my name is Karl Grenier. So I'm from Montreal. I've always been interested in neuroscience and neurodegeneration. It really goes a long way back. My bachelor, I was studying prion diseases, and then I went on to do a PhD studying Parkinson's disease, and I was working both on cell models and mitochondrial dysfunction. And then I thought that wasn't enough, so I went on into med school, and I fell in love with pathology, and I'm continuing to research on neurodegenerative diseases. It's a bit of a holy grail for me. I hope we can find a cure one day for these diseases. And so this article that I've published with Phidias, that he was nice enough to let me publish with him, is really about this new technology called organoids. Pretty much organoids are a collection of cells in 3 dimensions that self-organized into an organ-like structure. And we'll talk a bit more about that, I guess, later. And so the use of this technology in pathology and in medicine in general is about to explode. And here in this paper, we describe the reasons why, and mainly focusing on neurodegenerative diseases, because we think these organoids are really suited for this type of diseases, for its study, but even for more than that, for prognosticating and as a drug platform. But there are some challenges and some things that aren't quite perfect yet. And we talk about those limitations and how we can turn them into advantages. So it really is a paper for scientists, but also for pathologists or for clinicians or anyone who's interested about, a really cool new technology.
Okay, that's great. Just for our audience, why don't you tell me first, what you're the expert. Why are neurodegenerative diseases so important in society? Why is it a problem that we should pay special attention to?
Sure. So, well, first of all, I mean, on a personal level, I think, neurodegenerative disease are they're very hard for people and their loved ones. They're progressive, they're relentless, and they happen with age. And these are diseases like Alzheimer's or Parkinson's. So people lose their movement, lose their memory, their ability to think. So it's really awful diseases. But so, as I said, they're associated with age. So what's happening in our society right now, particularly in North America, is that we're getting better and better to keep people alive. So people get very old, but at the same time, we have no cure at all for these diseases. So we see many more people having these diseases, and yet we have absolutely no cure. So it becomes very important to study them, because we want to have, what's the point of living old if you're going to have one of these very debilitating diseases, And so it's bad on a personal level, but also economically, it's terrible.
Yeah, it's gonna be interesting to see how the life expectancy changes as we tackle heart disease and cancer. Exactly. It's scary to believe that this is a disease that could affect a large majority of living people. So we understand why it's a big problem in our society. What's missing from research that we've made so little progress over the last few decades?
Yeah, so this is really, the key point to get into our paper. And like, I personally have a real frustrating experience with that. I mean, I've done my PhD in Parkinson's disease, and I was working with cell models, right? So they're two-dimensional. So we had some human cells, so at least they were the right organisms, but they're very monomorphous. It's the same type of cells, and it doesn't reproduce at all the architectural or even the interactions that we have in a complex organ like the brain. Like, I mean, the brain is extremely complex. And on the other hand, you have animal models, which I've also worked with. So, you know, these animal models, first of all, well, they're not human, right? So they don't have our DNA. So they, you know, there are some proteins that are fairly well conserved, but, you know, it's not exactly the same. But I think even more important is that these neurodegenerative diseases have evolved into humans that live a lot longer. And so our cells in the brain, the neurons that die in these diseases, are subject to a lot of toxic molecules and DNA damage over time. And they've evolved to have mechanisms to palliate that, right? And we hypothesize that when these quality control pathways go wrong, then those cells die. Well, I don't think the rodents ever evolved those quality control pathways as well as we do for such a long time. So I think, you know, these models were good to learn a little bit, but they were going to fail us at some point because they don't recapitulate the brain and the lifespan of humans well enough.
That's great. That's great. So where do you see organoids fitting into the picture?
So organoids really come in perfectly to tackle these limitations. So first of all, they are human. So more than being human, we can take cells from a patient and grow them into their own organoids. So not only they have human DNA, but they have the human DNA of the patient. And second of all, so they grow into a structure which is very similar to our brain. In fact, it's about similar to the brain of a second trimester fetus, okay? But we can see structures that we would normally see in the brain. So we can really start to see how those neurons degenerate and how the process starts, because that's been the long-standing problem in pathology. You know, the brain is in the skull, and you know, we don't want to go biopsy there all the time. So, and especially when there's no clinical symptoms yet. But now we're able to see something that's similar to the brain without having to go inside the brain. We can just take cells and grow a mini brain.
Yeah, that's it's quite an intriguing technology, and I think one thing that I learned from this collaboration is really the expertise you have in overcoming some of the limitations that perhaps exist in current organoid models. For example, you said these structures, although they have some advantages, such as having real human-like structures like ventricles and layered cortical tissue, one of the major disadvantages for studying these types of diseases is their relative immaturity. And before we started working on this together, I was very skeptical how this technology would ever transpire to help address diseases of not neurodevelopment but neurodegeneration. And I think your expertise from your PhD provided a lot of insight and inspiration for this review that we wrote. So can you summarize that for our audience?
Yeah, so yeah, that's extremely interesting. In fact, is that you're right. So when we grow mini brains or cerebral organoids, these are equivalent to about, as I said, the second trimester of a fetus. So these are really primitive cells, and people will say, how can you compare that to an aging brain? Well, the fact is, because they are young, it actually allows us to make them age, and to see the whole process of how the damage accumulates in a very short span. So we can actually turn this into an advantage. And there are multiple ways to do that. So one of the ways would be to engineer those organoids to have diseases that we know of that accelerate the aging. And we know a few of these, what we call progeric syndromes. So some of the unlucky humans have those syndromes and they age very prematurely. So we can make these organoids have these diseases and they will age faster. And the second way to really make those organoids age is that, you know, over time, we've learned a little bit about aging, and we now know that it's a combination of damages to the cells, including DNA damage and mitochondrial dysfunction. So we can push that on the organoids and see them age, and then we can compare an aged organoid to a young organoid and see what are the effects of, let's say, a mutation that's associated with Parkinson's disease. So in fact, you know, it really becomes an advantage because we can condense time and study it. We don't have to wait 60 years to know what happens, really.
Yeah, that's truly amazing, and I think these types of synergies where we take the advantages of this emerging model system and we couple it with you know, expertise that people like you have is going to be really exciting. And I hope we continue to work together. Hopefully this is just the beginning, and I'm excited to see where we can take this.
Well, yeah, for sure. And I mean, your expertise too, right? And so, you know, these are complex little things, right? So we're used, you know, for example, in pathology to see two-dimensional sheets of cells and to analyze that. And now we have these very complex structures that resemble the brain. And so I think, you know, to analyze the whole areas of protein, so the proteome, and, you know, to analyze as well with mass spectrometry, or even to have, you know, deep learning to help us figure out what are the changes in these organoids will be key. And that's something I know you've been doing in your lab. So it really all comes in together, right? It's the perfect substrate for all these new capacities that we have.
Yeah, and a kind of shameless plug to our young trainees watching. I think I think as we're able to develop complex tissue, training in microscopy and pathology is going to be kind of revitalized in this new era where we not only can look at patient tissue, but we can generate artificial tissue in large amounts in a scalable way. And we need people with microscopic expertise that understand tissue site architecture to really look at these and you know, and tease apart key subtle architectural anomalies that could you know, hopefully guide new treatment approaches.
Right, and once we identify these anomalies, you know, that we can see by a pathologist, now you have the possibility to take skin from someone that doesn't have the disease, and maybe you'll see those anomalies in advance, and for the first time practically ever, you'll be able to enroll people in clinical trials that are, you know, predicted to develop neurodegenerative disease. And that's huge, because all of the drugs that we've tried in clinical trial for neurodegenerative disease have been tried on people in which the neurons are already dead and they're gone. So, you know, your drug has a high chance of not working. So I think this is really where it could hit its maximal potential.
Amazing. Anything before we let you go? I know you're a busy guy, you're a new father. Anything else you want to tell the channel?
Well, I would say stay curious. And, you know, these diseases are tough diseases, but there's a lot of smart and hardworking people trying to find a solution. And if you're one of them, well, I would suggest you to read our paper. You might get some ideas, and who knows, might collaborate one day.
Yeah, that's very true. One last request. Can you tell the audience to like, subscribe, turn on notifications, and tell their family and friends about our channel?
Absolutely. I advise you all to do that. So please like, please share this and yeah, like the YouTube channel, and that would be greatly appreciated.
Okay, Dr. Grenier, thanks for your time.
No problem.
And keep in touch.
Yeah, great to see you.